Before you will be able to determine which are the bad or good bacteria, you must identify the type of bacteria found in a particular sample. Over the past century, microbiologists have searched for more rapid and efficient means of microbial identification. The identification and differentiation of microorganisms has principally relied on microbial morphology and growth variables. Advances in molecular biology over the past 10 years have opened new avenues for microbial identification and characterization. Methods of bacterial identification can be broadly categorized to phenotypic techniques based on profiling either an organism's metabolic attributes or some aspect of its chemical composition, and to genotypic techniques based on profiling an organism's genetic material, primarily its DNA. Genotypic techniques have the advantage over phenotypic methods that they are independent of the physiological state of an organism. They are not influenced by the composition of the growth medium or by the organism's phase of growth. Phenotypic techniques, however, can yield more direct functional information that reveals what metabolic activities are taking place to aid the survival, growth, and development of the organism. This may involve, for example, in a microbe's adaptive ability to grow on a certain substrate or in the degree to which it is resistant to a cohort of antibiotics. Genotypic and phenotypic approaches are complementary and use different techniques. Phenotypic characters of bacteria include microscopic morphology. Morphological characteristics include colony morphology such as color, size, shape, opacity, elevation, margin surface texture, consistency, and so on. These characteristics are observed after the incubation period in the cultures on the solid media. In liquid cultures, we can observe the pellicle formation and sediment formation. Gram staining depends on the differences in the thickness of a peptidoglycan layer in the cell membrane between gram-positive and gram-negative bacteria. Gram-positive bacteria with a thicker peptidoglycan layer retain crystal violet stain during the decolorization process, while gram-negative bacteria lose the crystal violet stain and are instead stained by the safranin in the final staining process. In this video, you will see the different steps involving gram staining. This involves three basic steps: staining with a water-soluble dye called crystal violet, decolorization, and counter-staining, usually with safranin. Label the microscope slides. Mark a circle at the middle of the slide. Using a metal inoculation loop, sterilize by flaming. Using a septic technique, get a drop of water and place it on the slide inside the mark. Flame the metal loop again to sterilize. After allowing it to cool, get a colony of bacteria from an agar plate culture and transfer it to the slide. When finished, flame the loop to sterilize. Let the liquid dry for 10 minutes. Heat fix the bacteria by passing the slide over the flame 3 times. Add crystal violet stain. Then rinse the slide in distilled water. Add grams iodine and rinse again in distilled water. Add a gram stain decolorizer and rinse again with distilled water. Counter stain using safranin. Rinse with water. After the slide is completely dried, observe the slide under the microscope. Biochemical tests are among the most important methods for microbial identification. Routine biochemical tests include tests for catalase, methyl red, citric acid utilization, and hydrogen sulfide production. Microbial biochemistry tests shorten the time required to identify microbes, reduce cost, and ensure or enhance the accuracy of identification of an unknown sample. It is the fastest developing trend in microbial identification. In recent years, the rapid commercial test kits for anaerobic bacteria have become available. One of the kits available is the EnteroTube. The EnteroTube is an example of a rapid, multi-test system used in identification of unknown oxidase-negative, gram-negative, rod-shaped bacteria of the family Enterobacter. It consists of a tube with a flat side and contains 15 compartments for different biochemical tests. First, label the tube. Remove the caps at both ends. Point the end with the green agar downward. Scrape bacteria onto the tip of the inoculation wire as you rotate the tube. Inoculate the agar wells by pulling the wire all the way out and placing the wire back into the tube. Break off the wire at the indentation. Replace the two caps. Then use the tool to poke the holes in 8 U-shaped channels on the side of the tube. Then incubate the tube. The results pad and color reaction chart permit a rapid check of the positive reactions obtained with EnteroTube. The checked positive numbers are totaled and the composite number is then located in the interpretation guide to identify the organisms. When 2 or more organisms are listed, the confirmatory tests required to further identify them are also given. The protein and polysaccharides that make up a bacterium are sometimes characteristic enough to be considered identifying markers. The most useful of these are the molecules that make up surface structures, including the cell wall, glycocalyx, flagella, and pili. Enzyme-linked immunosorbent assay, or ELISA, is an analytical technique to detect the presence of an antigen or antibody in a given sample. It shows wider applications in clinical diagnosis, in pathological studies, and in quality control studies. Virtually all microbial species have unique antigens, and such type of antigens can be exploited as specific molecules of detection by ELISA. The variations in ELISA allow us to detect either antigen or antibody, identifying the different strains of microbes at a time, and also in characterization of the epitope distribution of microbial surface. Commercially available ELISA test kits include microplates coated with the antigen of interest and all the necessary reagents. Dispense the diffusion buffer provided by the kit into the wells of the antigen-coated microplate. Add the positive and negative controls provided by the kit. Then add the samples in the remaining wells. Incubate the microplate at 37 degrees for the time indicated to permit the development of an antigen-antibody immunocomplex. After incubation, wash the wells by rinsing with the wash solution provided in the kit. This washing step is repeated several times. Prepare the enzyme-conjugated antibody. There are two types of ELISA, direct and indirect, which differs in the antibody use. Dispense the conjugated antibody into each well. A second incubation is carried out to allow development of a second immunocomplex. After the incubation, the entire washing procedure is repeated to eliminate the conjugate in excess. Finally, add the substrate and allow for color development for a few minutes in the dark. When the color reaction is finished, add the stop solution and read the results using a spectrophotometer at the recommended wavelength. Positive results are usually colored. The optical density of the color is measured by means of a spectrophotometer. Another popular method of bacterial classification is through characterization of the types and proportions of fatty acids present in the cytoplasmic membrane and outer membrane. This technique is nicknamed as FAME, or fatty acid methyl esters analysis. The fatty acid composition of prokaryotes can be highly variable, including differences in fatty acid length, the presence or absence of a double bond, rings, branch chains, or hydroxyl groups. The fatty acid profile can help to identify a particular bacterial species. FAME is in widespread use in clinical, public health, and food and water inspection laboratories, where the identification of pathogens and other bacterial hazards needs to be done on a routine basis. Recent years have seen an explosion in the development and application of molecular tools for identifying microbes and analyzing their activity. These tools can be divided into those based on nucleic acids and other macromolecules, and approaches directed at analyzing the activity of complete cells. Fluorescent in situ hybridization, or FISH, is increasingly used to observe and identify intact microorganisms in environmental samples and clinical samples. By using a probe that binds to certain ribosomal RNA or rRNA sequences, either species-specific or groups of related organisms can be identified, and characteristics of rRNA can be studied that make it ideal for classification. In this video, you will see the different steps for fluorescent in situ hybridization, or FISH. You need to prepare the sample slides, the samples must be denatured with heat or chemicals. This denaturation step is necessary in order for new hydrogen bonds to form between the target and the probe during the subsequent hybridization step. The probe and target sequences are then mixed together, and the probe specifically hybridizes to its complementary sequence on the sample. If the probe is already fluorescent, it will be possible to detect the site of hybridization directly. In other cases, an additional step, adding DAPI stain, may be needed to visualize the hybridized probe. Hybrids formed between the probes and their chromosomal targets can be detected using a fluorescent microscope. PCR or polymerase chain reaction can be used to amplify a specific nucleotide present in nearly any environment. This includes DNA in samples such as body, fluids, soil, food, and water. This technique can be used to detect organisms that are present in extremely small numbers as well as those that cannot be grown in cultures. The most commonly used DNA sequence for bacterial phylogenetics is the highly conserved 16S rRNA gene sequence, and primers have been designed to selectively amplify bacterial 16S rRNA genes. This method is the focus of the Bacterial Identification Virtual Lab.